So our final speaker for this morning is one of the pioneers in molecular imaging and spectroscopy. Professor Shimon Weiss is the Dean Willard Chair in Chemistry and Biochemistry at the University of California, Los Angeles. He was the first to introduce the single-molecule FRET method, as well as using quantum dots for biological imaging. Professor Weiss' cutting-edge research focuses on novel detectors for advanced imaging and spectroscopy and developing new optical imaging techniques and tools for single-cell physiology. Today, it is our pleasure to have Professor Weiss share with us some of his exciting developments in optical molecular imaging for the study of molecular machines and molecular interactions in vitro, in live cells, and in small organisms. Professor Weiss, please.
So, good morning, everybody, and I would like to thank the organizer for inviting me. I would like to repeat what Atsushi Miyokari said. I tremendously enjoyed this meeting. I think it really paints the future in bright colors. What I'm going to discuss today is a few examples of molecular tools based on fluorescence. And that will be my tool. That's what we do in my lab. We develop several different tools. And fluorescence, we have heard quite a bit about fluorescence today, but let me just remind the young audience here in the hall. We use molecules in food coloring. You are probably familiar with gummy worms and other types of food that are colored. These molecules are usually composed of several different benzene rings, and they have very interesting optical properties. Using quantum mechanics, we can calculate the energy levels for such molecules, and this is what is known to be the Jablonski diagram that basically represents different energy levels for such molecules. And we are going to consider, for the sake of this presentation, these molecules basically to be light bulbs. So I'm going to talk to you about light bulbs and how we use light bulbs to explore molecular frontiers. So I'm going to tell you 4 different stories today. The first story will be single molecule fluorescence resonant energy transfer. The next story is going to be about quantum dots. And then I'm very briefly going to touch about super resolution microscopy. This is really the next frontiers in imaging, and the founders of this whole new era are going to talk to us today in the afternoon session. So I'm not going to talk much about super resolution, just demonstrate one method that we developed following the giant steps taken by Stefan Hell and Eric Betzig to simplify things and make things faster. So we'll touch upon that, and eventually I'm going to finish talking about some new tools developed with my friend and colleague David Ben-Simon. We heard a lot about reading and writing the genome, and I think one of the important future steps will be to read and write phenotype as well, so that will be this topic. All right, so let's go on. Single molecule FRET, fluorescence resonant energy transfer. So think about this light bulb that we have. Let's call this one yellow light bulb a donor molecule. And if we are going to bring another light bulb in the color red here, we will call that molecule an acceptor molecule. Those 2 molecules, if we bring them close enough together, are going to interact with each other. So how do they interact with each other? You guys grew up on this generation of devices. I actually grew up on this type of devices. In both cases, these 2 devices rely on dipole-dipole interaction. What is dipole-dipole interaction? This is our transducer. It's a radio station or it's a cell phone tower. We basically accelerate electrons in the antenna. The antenna will radiate electromagnetic radiation, and this electromagnetic radiation is picked up by another antenna on the receiver side. It works for cell phones. It works for iPhone, this device here, and it works for the transistor that I grew up on as well. So we basically, by accelerating electrons in one antenna, we generate electromagnetic radiation. This is our oscillating dipole, and that will basically induce acceleration of electrons in the receiving antenna, and this is another dipole, and we have here dipole-dipole interaction via the magnetic field, electromagnetic field. This dipole-dipole interaction is dependent on the distance, and we'll talk about this in a second. Basically, for the cell phone or for the transistor, we are going to modulate this electromagnetic radiation with some content that will be our audio that we put on top of this electromagnetic radiation, and then we'll basically be able to demultiplex this signal and read it out on the receiver side. So this is dipole-dipole interaction. This happens in the electromagnetic radio frequency range, but if we do the same for molecules, we will still have the exact same interaction. Here we have the donor and acceptor molecules now described in this Jablonski diagram as I described before. We have here a donor molecule, we excite it with a laser light, and this molecule can relax by emitting photons. Those will be green photons. But if we bring another acceptor molecule very close by, instead of relaxing and emitting photons, by dipole-dipole interaction, we can excite this other molecule. And how much is being excited, the theory of which was worked by Furster in the late 40s of the previous century, is dependent, basically, the rate of this transfer of energy depends on 1 over the distance to the 6th power, or the efficiency of the energy transfer between donor to acceptor, scales like 1 over 1 plus the distance to the 6th power. This is a very, very strong distance dependent. Instead of doing this in the RF region for the radio frequency, this is done in the optical range, and therefore the range of this dipole-dipole interaction in the optical range is much, much shorter. It's very effective on the range of, on the order of 2 to 10 nanometers. That is molecular scale distances. So the concept is very simple. Basically, you bring those two molecules together, you excite the donor, and then you're going to analyze the spectrum coming from the pair. If the donor and acceptor are far apart, you'll see mostly emission from the donor. If the donor and acceptor are very close to each other, you see mostly emission from the acceptor because energy was transferred to the acceptor. As I said, the theory was worked out by Forster. First demonstration that you can actually use this method to measure distances was done by Stryer and Hagoland in 1967, and from that on, this method has become a really important tool in biochemistry and cell biology to measure interactions, to measure distances. However, one important point is that if you do this measurement as was done from 1967 until 1996, you do those measurements on ensemble, what you see here is a simulation of many pairs of donor and acceptor. They are tethered here, as you can see, and there is some fluctuation in the distance between donor and acceptor. And you excite all of the molecules in your test tube. You collect all of the green photons onto one detector, all of your red photons into the other detector, and basically you get those two signals. The ratio gives you basically one number, the average number of the distance for all those pairs. If you reduce the number of molecules in your test tube, now you get much noisier signal. You can note that the signal between donor and acceptor is anti-correlated. When one goes up, the other one goes down. The range of values that you get is much broader now. You get a histogram. And if you further reduce the number of pairs into a single pair, you get very strong fluctuations. And basically, over time, you'll see here only one value. So you get the ability to actually measure changes in the distance that you couldn't do if you measured traditionally over the ensemble. This has basically quite profound implications because what it means is we can move from protein crystal structure into things that are changing over time. So this is, for example, folding of a protein. This is the actual motions within the enzyme F1, F0 ATPase. It's a rotary motor. And by taking the idea that we now can measure distances as function of time, changes of conformations as function of time, we can actually go do specific chemistries. We heard this morning about specific chemistries. If we can put a donor and acceptor molecules on particular sites on those molecules, we can see how things move. So we're taking our molecular machines that basically do all the chemistry and the metabolism inside the cell, and we can look at moving parts on the nanoscale, on the molecular level, using this method. So we proposed quite some time ago, after introducing this method, that we could do all kinds of different analyses. For example, think about all kinds of DNA-based enzymes that process DNA for replication, for transcription, and so on. You can imagine to label the enzyme and look at the Pacman basically working on that DNA template, and by measuring the signal for the donor and acceptor, we can measure about the enzymatic activity. We can do the labeling in all kinds of different positions. So that was labeling of 2 sites on the protein itself. You can also measure the translocation of the enzyme with respect to the substrate DNA. If you label the DNA, this is called the DNA encoder that was recently introduced by Everett Lipman. So if you are able to label your molecules properly, you could actually really look at moving parts. And by now, there are hundreds of, if not thousands of groups that utilize this type of method to basically take structural biology into dynamic structure. Specifically, I'm going to give only one very brief example of what we have done in my group. So you heard earlier yesterday about the central dogma in biology. All the genetic content is residing within the DNA. The enzyme RNA polymerase basically translates this information into messenger RNA, and the messenger RNA is read by the ribosome to produce proteins. In my group, we study this transition from DNA to RNA through the enzyme RNA polymerase. I'm going to give you one example of how we use single molecule FRET to answer questions that could not be answered by any other method. So for the enzyme RNA polymerase to make a new messenger RNA, it needs to find a region within the genome which basically designates the start of transcription for a new gene. This is called a promoter. The enzyme binds the promoter, and through a series of steps, it will basically form first what is called the closed complex. Through some isomerization step, it will open up a transcription bubble. Basically, you all know about the double helix. The enzyme basically has arms. It's going to separate the double helix and basically create some kind of a bubble that is about 11 to 15 bases long. This is called the open complex. And if at that point you provide the enzyme with its fuel are the nucleotides that make the RNA, it will start to transcribe. Now, what is very interesting about this first initiation of that process is that the enzyme is, by statistical mechanics, start to make some transcript, lose them, march back to the beginning, to the start of the gene at the promoter, try again, loses the transcript, slides back, try again, and so on. This is called abortive initiation. Think about an old car going up a hill and the engine shuts off, it slides back, it try again, try again. Somehow if it picks up some thermal energy, it can go over the hill, and at that point the enzyme is going to be committed to go all the way to termination side and make the full message. At that point it will be very processive, called, that will describe the elongation step. So during this abortive initiation, one can detect actually the synthesis of many short transcripts going between 2 and 8 bases long. And you can actually artificially lock the enzyme in this state. If you make the first 8 bases of a message containing only 3 bases out of the 4 nucleotides, the enzyme will never cross the hill and go to elongation. And during that process, you can actually perform some really interesting experiments. So my colleague at UCLA, Jay Gralla, did some very important experiment in the early 80s that could not be explained for over 30 years. His experiment basically measured the footprint of the enzyme on the DNA. So think about the enzyme sitting on the promoter, and basically we can cross-link the enzyme to the DNA, cut all the free DNA, and then collect the protein and the part of the DNA that is protected by that protein, extract the DNA and analyze the DNA. And with a surprising result when he did the experiment for the abortive initiation part versus the elongation part was that the protection, basically the footprint of that enzyme over the DNA is different in initiation compared to elongation. And that difference was a subject of intense study for 30 years. 3 different models came to explain that difference. The first model is called the inch-warming model. And the inch-warming model says that the enzyme works like a worm in the initiation part, but is not working like a worm in the elongation part. And what that means is this protein structure changes in the initiation part. Basically, the leading edge of the enzyme is stretching forward and backwards when it makes those short transcripts. So we could test using the single molecule FRET to see if indeed the enzyme undergoes conformational changes in the initiation part. Another model doesn't require changes in the protein structure for initiation. Instead, it assumes changes in the DNA structure. As I told you, the enzyme opens the transcription bubble, and then it needs to reel in the template DNA, the message that is going to transcribe to messenger RNA. And that means that you change the dimension of the bubble, and if we label the DNA 2 sites, we can actually measure conformational changes in the DNA. The last model that came about says that you don't need changes in the protein or in the DNA. It's enough to think that the enzyme sits most of the time in the back position, make very brief excursion, and then comes back and again sit for a long time in the back position. And that can be tested by putting DNA upstream of the promoter and, putting a dye and then putting a donor dye on the backside of the enzyme and see if there are translocations during the abortive initiation. The experiment that we did and published in 2006 came up with really clear results. There was extensive work done from the early 80s until 2006, and this basically problem could not be resolved. And the reason is that you cannot take an ensemble of many enzymes and synchronize them, all of them to do the same thing at the same time. And only the single molecule method allows to give the answer. So basically, we did exactly what I mentioned. We labeled the DNA upstream and the backside of the enzyme. We locked the enzyme in the abortive initiation cycle, and basically, you cannot see here because of the projector, but there are 2 histograms here for basically measuring if there is any translocation between the enzyme and upstream DNA. And what you find is that there is no change, so basically we can rule out the transient excursion model. We are left with 2 models only. The next experiment is to label basically and test for changes within the protein. So we want to see if the protein changes its structure, and we have 2 dyes in the right positions, and we compare again. Again, you cannot see the histograms, but basically we can find here no change, and therefore this model is not working. So we are left only with the with the third model, and that might be the most correct model, but let's test for that model as well directly. And this is shown here. So this model basically says this is the scrunching model where the enzyme pulls in the DNA and the DNA changes its structure. So we label the DNA 2 sides, and indeed we see clear change in distance on the DNA, and we can point to the fact that the scrunching model is the correct model. So that was one example. There are many, many examples for using the method, but we talked a lot about the central dogma and about various enzymes. I just wanted to show the power of single molecule FRET. All right, the next technology is quantum dots. And Sarah Croto told us about quantum mechanics and encouraged young people to go into quantum mechanics. So let's talk a little bit about quantum mechanics. I already described Jablonski diagrams. Let's talk about semiconductors. The equivalent of Jablonski diagrams for semiconductors are basically the band structure of the semiconductors. This is the valence band, and this is the conduction band. Basically, we have a parabolic dispersion relationship, and valence band is equivalent to the HOMO levels in organic molecules, and the conduction band is equivalent to the LUMO. Let's shine laser light and promote an electron from the valence band into the conduction band. This is our photon. we are going to leave one lack of electron behind in the valence band and put this electron in the conduction band. This lack of electron here is called a hole, and we have an electron here. Those are quasi-particles within the semiconductor. And if we have now a positive charge and a negative charge, we have something that looks very much like the hydrogen atom. Therefore, this entity is going to form something like a hydrogen atom within the semiconductor. We have electron orbiting around the hole. This is another quasi-particle called the exciton. And this exciton resides within the crystal structure. Again, you miss all the details here on the slide, but basically the cyan points here represent lattice points on the crystal, and this exciton resides over several unit cells within the semiconductor. There is a typical radius that is called the Bohr radius that tells you how far apart the electron is from the hole. And if we now take this exciton and we put them not in large bulk semiconductor, but in very small particles called quantum dots, nanoparticles, then the fact that the size of the particle is similar to the size of the exciton brings about quantum confinement effects. So let's talk a little bit about quantum confinement. This is a simple particle in a box problem studied in first-year quantum mechanics in college. Basically, what you see here is a box. Here at the bottom, you see the wave function of the electron residing within the box. The green line represents different energy levels. And if we change the box size, you can see that the number of confined states is getting smaller and smaller, and the energy spacing between different confined states is getting larger and larger. What it means is that if we now just a minute. All right. If we now take our exciton and we are going to confine it in a smaller and smaller size quantum dot, we are going to have this particle in box squeezing the size of the box. The energy levels are going to change. This is called quantum confinement. So if you have the exciton in a large quantum dot versus the exciton in a small quantum dot, the particle in the box picture is going to look like this. For the smaller particle, the energy spacing is larger and the band gap gets larger. Basically, we are going to have a larger band gap for a small quantum dot versus a large quantum dot. This can very easily be implemented by chemistry. This is basically describing the chemistry of quantum dots. This was developed by Lou Bruce and later on by Paula Levisatos and Munjibo Wendy. You put a hot soap solution basically in a flask. You heat it up for 300 degrees and you add metallo-organic precursors that are going to break the bonds with the organics, and basically inorganics are going to nucleate and grow into perfect crystalline material. And the key point here is that all of the quantum dots grow at the same rate to have uniform size, and you can control the size by controlling the duration of the reaction. If the reaction is long, you are going to have large particles. If the reaction is short, you're going to get small particles. And therefore, we can have a classical example of particle in a box and change the box size and change the energy levels. What that means is that we can actually have different color emitting particles with very unique spectral properties that are different from conventional organic dyes that are used in fluorescence. So this is shown here. You can actually excite all of those different size particles with single laser and get all these colors out at the same time. This is something very desirable. Another very important property of these particles, they are very, very stable compared to organic dyes. If you take an organic dye and shine light on it over time, the organic dye is actually going to decompose and stop emitting light, while quantum dots stay bright for a very long duration of time. You can use many different materials to make such quantum dots. Within each material, you can change sizes, so you really can cover the whole optical spectrum with many different sizes and many different compositions to get all kinds of different color quantum dots. So, as I mentioned, there are many, many different good properties for these quantum dots, and they are bright. They're very, very bright compared to organic dyes. They have many colors, and this is a list of all the good properties. I'm not going to go through that list. And what it really means for biology is same way we use the internet to send many, many different messages by encoding different messages effectively with different colors through optical fibers, we can imagine to do the same for biology. So this is a picture taken from a review by Roger Tsien, painting different proteins in the cell with different colors, combination of organic dyes and quantum dots and all kinds of things. The unique property of the quantum dots now is that they are very stable, very bright, and we can see individual ones even with our own naked eyes. So what it really means is the following. So I don't get the audio here?
Ball game.
The machine works? Do the kids know?
Well, yeah, the kids know.
That's great!
It's not that great.
Why?
I shrunk the kids.
What?
And the Thompson kids, too. They're about this big. They're in the backyard.
What?
Through the... Well, with the...
So I don't know if you are familiar with that movie. This is called Honey, I Shrunk the Kids. And this is just to say that the quantum dots allow us to shrink the kids. We can actually go and paint individual proteins within the live cell and follow those individual proteins for a long period of time. So this is an example of one application that we pursued in my group, which is the organization of membrane proteins within the membrane of live cells. There are many, many different proposals and models. The main question is, are membrane proteins undergoing simple diffusion within the membrane, or they are organized in some fashion in some rafts, lipid rafts, or patches of different content lipids? And if they are, there might be significance for the way the cell communicates with other cells and with its environment. So the type of experiment one can do with quantum dots now is basically tag membrane proteins and watch them for a very long duration. So the red spot here represent individual proteins in the membrane of live cells. And you can see some of them are diffusing quite a bit. Some of them are localized and not moving much. You can take thousands of such trajectories, analyze them, basically characterize them by their diffusion constant, and very clearly you come up with bimodal distribution of fast-moving and slow-moving membrane proteins, and that supports very much the idea of phase segregation and lipid rafts in the membrane. So this is an example of what quantum dots are useful for. And I told you they are very bright, and they last for a long time. We can look at single molecules, and we can look at single molecules in extreme events. What you see here is a cell organelles, and you see individual organelles loaded with quantum dots, except that this is now not an experiment done in a tissue culture, but an experiment done basically in one cell in the whole context of a living animal, in this case zebrafish. So we uploaded quantum dots into the embryo immediately after fertilization, and then the egg divides and keep on dividing, and you get the full development of the embryo. And here you can see close to the tail individual cell labeled in a background of unlabeled cells, and we can see the organelles within that cell, bringing the idea of possibly performing single molecule experiments in the right context, in individual cells, when all the circuitry is wired up rather than the artificial arrangement in a tissue culture. And you can do many other things. This is an example of injecting quantum dots into the heart. There is a stage within the embryo development that you can do this, and the fish will survive. So you can put quantum dots into systemically into the blood system, and you can follow it. And if you functionalize the quantum dots, you can target particular cell types. And with some work, you can actually use the zebrafish as a way to screen the surface properties of those quantum dots such that you get very nice uniform distribution without aggregation, as you can see here in the top movie. Now, I talked about the good things about quantum dots. There are many disadvantages as well. To start with, they are relatively large compared to organic dyes. The surface properties are not perfect. They are sticky. The avidity, the number of, let's say, antibodies that we can put on them is not controlled very well. And they blink. So here you see a movie of quantum dots deposited on the surface of a cover slip. And if you take a movie as a function of time, you see that they come on and off. And this is basically not a good property because you don't get all the photons that you would like to. You could get even brighter quantum dots if they won't blink. And in the context of what I've shown you before, when we analyze trajectories within the membrane, that would also be problematic because the blinking will cause some loss of following up those diffusion trajectories and basically will make the life a little bit more complicated. But I want to take this lemon and convert it into a lemonade. And this is the Superresolution method that we developed recently. The idea is not my idea. The idea is your underlying idea. The execution was done in my lab. And what you see here, again, I think because of the quality of the projector, it's hard to see that this is a movie of a fixed cell now. So we are no longer looking at live cell. This is a fixed cell. And the concentration of quantum dot that we use to label the cytoskeletal, the microtubules, is very, very high compared to this single molecule regime that I've shown you before, where we have very, very few quantum dots in the field of view. So here the microtubule network is labeled with quantum dot, and you can actually see the blinking of those quantum dots. Okay, that was my timer. So taking this blinking, we can actually use a very simple mathematical algorithm to go from here into there. And again, the poor quality is not showing really the beauty of that picture. Here you see super resolution, you see contrast enhancement, and you see three-dimensional sectioning, all done with a simple mathematical algorithm that is really due to the blinking of quantum dots. And in fact, the same algorithm can be worked on blinking of organic dyes and blinking of fluorescence proteins. This is labeling of the microtubule network with quantum dots in live cells, and this is actually not simple to do. This is done by microinjection. And this is a cell line that we received from our speaker in the afternoon, Atsui Miyawaki. This is basically DROMPA, fluorescence protein fused to HIC5 in focal adhesions. And you can work out conditions, extremely simple condition, a wide field microscope, LED illumination. It's almost a simple lamp, no lasers are needed, one color, and you get the blinking that you need to get. And this is making this type of work very, very simple. So you can get super resolution, fast super resolution with a simple fluorescence protein fusion and a wide field microscope. All right, so the last topic, this is, we heard about reading and writing the genome. Let's talk about reading and writing phenotypes. And we heard the advantages of zebrafish. Zebrafish is transparent. We do fluorescence and optics, so we like transparent organisms. It develops very quickly. It lays about 400 eggs each time, so you have a large number and you can do statistics. And it's basically a really very, very nice organism as a model system to do optical work with. So here, within 24 hours, you get an adult fish that escaped the egg, and you can do all kinds of interesting experiments with. This is, again, the work that I'm going to present now is not my idea. It's the idea of David Ben-Simon, a good friend and collaborator, a group of collaborators in Paris who developed the chemistry for this work. We have done the imaging part. And our goal eventually would be to be able to write individual genes into single cell in a whole animal. For example, overexpress a particular gene or an oncogene in one cell at a particular point in time and space. What I'm going to present now is not yet that goal, but actually using a native endogenous molecule called retionic acid that is involved in the development of embryo or the development of the brain. So The idea is to use light to basically make a perturbation in a very local area, in a very temporal delta function or step function way, and then optically read out the effect of the perturbation. So this is reading and writing phenotypes. What we have used here is the fact that retionic acid can be isomerized by UV light. The cis-retionic acid is an inactive molecule. The trans-retionic acid is the active molecule. And people have shown in the past that during the development of the fish, if particularly you're looking into the development of the brain of the fish, like our brain, has lobes called rhombomeres. You can see here this is a normal development of the brain. You see rhombomere 3 and rhombomere 5. This is done by fluorescence in situ hybridization after sacrificing the animal. If you block with the drug the synthesis of retionic acid, you don't get normal development. You get only one rhombomere developed. But you can actually rescue this phenotype by adding retionic acid from the outside, and this is shown here. We gained a transgenic animal from our collaborator Shui Ling in UCLA, who basically did the same, except that now we have a green fluorescent protein in those two rhombomeres. And here again, inhibiting the synthesis of retionic acid, we don't get normal development. If we add retionic acid from the outside, we can recover the phenotype. So here you see development. Now we follow with fluorescence the development of the brain. And here we inhibit the synthesis of the native retionic acid, and you see that mostly only one rhombomere grows up. But if we supplement from the outside, after inhibition, we add retionic acid, trans-retionic acid, we get normal behavior of the brain. Now, we can add, instead of adding trans-retionic acid, we can add cis-retionic acid that is not active, but we can activate it with light, locally and temporally. So here what you see is we do this experiment and then we use UV light and we illuminate either the tail region or the head region, and you can see that basically this is illuminating the tail. Basically the brain doesn't develop properly, and illuminating the head, we release the cis-trans into trans-RA, and now we can get full recovery of the phenotype. And the idea is now that we can go and test developmental biology models where we can work against known gradients of the small molecule and build a gradient at will and see how the animal develops if you basically perturb the concentration gradient of this morphogen called retionic acid. We would like to take this type of ideas further. It's very much following what has been done in the ion channel field with bacteriorhodopsin. This is basically optogenetics. We would like to be able to do this for any gene, and in particular for oncogenes. We would like to start cancer very controlled way in a single cell within the embryo. So work is ongoing in this area. I would like to conclude with thanking all my student, postdocs, current and former, and all my collaborators and funding agencies, and you for your attention. Thank you.